Now that we have examined structures of the external, middle, and inner ear, or internal ear, I want to focus on what can be tested under a microscope or from a PowerPoint question. So to review the pathway of sound, I've just briefly sketched a schematic. So right here we have our tympanic membrane, separating the inner from the middle ear in orange. This entire structure, this kind of snail-shaped structure, is going to be our cochlea. Kind of looks like a conch shell. That's our cochlea. And then in between we have our 3 ossicles. So it's not a super pretty sketch, but it does the job. And then in green I have shown the pathway of sound waves and how they're translated from a mechanical sound wave into a sensory signal. So sound waves are going to hit that tympanic membrane. They're going to travel through our 3 stapes, our three ossicles rather, and we have the incus, malleus, and the stapes. And then there's an important structure to point out here. We have 2 windows to the cochlea. So window number 1 and window number 2. Window number 1 is our oval window. This is what lets sound waves into the cochlea. And we need window number 2, the round window, which allows vibrations in opposition to the direction of sound. So this stuff can get really complicated really quickly, but basically this round window is going to vibrate and oppose the sound waves coming in, the auditory signals, and the existence of these 2 windows is what allows our fluid within the cochlea to move around. And that's really important because that movement of the fluid is what actually allows a sensory signal to be taken to the brain and the central nervous system. But before we get to that, let's finish this pathway of sound. So it's going to enter through the oval window at number 1. And then this sound is going to take this really twisting, tortuous path. It's going to go up, wrap around, and then hit that round window. And all along this pathway in between, there's going to be fluid moving back and forth within what we call the membranous labyrinth. So this inner portion here that's outlined by the dotted lines, so this entire section inside, around which the sound waves are wrapping in this image, this is all filled with a liquid called endolymph. Let's write that term over here. Endolymph. If we think about lymph nodes or our lymph system, lymph is a type of fluid. So in this case, we have this inner fluid. Endo means within. So this is fluid inside that inner structure called the membranous labyrinth. So keep that term in mind for structures of the cochlea. Additionally, we have some nerve fibers here in purple. So these fibers wrapping around the cochlea and then joining to make this nerve, these are part of our auditory nerve, sometimes called the vestibulocochlear nerve. And right now we're looking at the cochlear part of that nerve that's innervating the cochlea, and that's what is going to take the output from these receptors back to the brain for processing. So let's focus on the cochlea right now. So I mentioned the membranous labyrinth. That's going to be that fluid-filled inner portion that we've outlined in purple here. In addition, we have a bony labyrinth. So if we keep up with that shell analogy and think of the cochlea as a snail shell, then the bony part, the outside part, is going to be bony labyrinth. And this is filled with a different fluid called the perilymph. And it's through this fluid that our sound signals are going to travel, bounce off that round window, yada, yada, yada. So try to keep these terms separate. Bony labyrinth goes with perilymph, that outer lymph or fluid. And then our membranous labyrinth outlined in purple is going to be filled with that endolymph, that inner fluid. Additionally, we divide the cochlea into 3 different chambers, and this is where this kind of sketch becomes less useful because it's hard to imagine 3 chambers when we're just looking at the snail shape. So this is the point where we want to look at histology, and boy does this look different than that image above. So what we have done is we've basically zoomed in on a really tiny part of the cochlea. If you think about this as a snail shell, we're basically just picking an arbitrary point, maybe here. And we're cutting into it, and we're looking at a cross-section now. So it's a different perspective, and this is the cross-section that we will see. So let's get ourselves oriented here. We have 3 chambers, so let's go ahead and label them here. These are our 3 chambers, or scala. We're going to use that term. It means chamber. But whenever we are talking about the cochlea, we use the term scala. So we have 2 chambers that are going to be associated with that outermost bony labyrinth filled with perilymph. And then we're going to have 1 inner chamber, or scala, that is associated with that inner membranous labyrinth. And if we just look at this image here, we kind of do have 2 outer chambers. So 1 and 3 are sandwiching chamber number 2. And so you would be correct in thinking that 1 and 3 are part of our bony labyrinth, and then our middle chamber, or scala, is going to be part of that membranous labyrinth, that inner portion. So let's go ahead and give these guys names. So once you've oriented yourself, we have 2 other structures we can use to figure out which chamber is which. But number 2 is going to be really easy. 2 is our scala media. It's our middle chamber. That one's usually pretty easy to identify because it's always sandwiched between the other 2. We have our actual auditory receptor, which is going to be this structure here. We'll get to that in just a minute. And then we have an additional membrane. This is a dividing membrane separating chambers 2 and 3. And this membrane is called our basilar membrane. And these are the 2 landmarks you want to use to identify your chambers. So from here, I'll just tell you scala vestibuli is separated from the middle chamber by this basilar membrane. So this tells us that 3 is going to be our scala vestibuli. There it is. And then our remaining chamber, number 1, is scala tympani. So I don't have a great memory cue for this one. You're just going to have to recognize that we have a membrane here spanning the cochlea, which is called our basilar membrane, and that is separating scala media, our middle chamber, from our scala vestibuli. And then by process of elimination, the largest chamber we see here is going to be our scala tympani. Now let's focus in on this structure. This is our actual auditory receptor. So this whole cochlea is helping to get sound waves circulating so that the disruption of fluid is going to activate this auditory receptor. And this is called our organ of Corti. And you're going to hear me say over and over again, find the shrimp. To me, this structure looks shrimp-like. It looks like a shrimp. So this is a term I use to help orient myself when I'm looking at a perspective like so, or the slide above. So I always go to this structure first. I find the shrimp, and then from there you can identify all of your scala. You can find your basilar membrane as well. So that's all that phrase means. So we've zoomed in now. Spanning the cochlea, we have this organ of Corti. And I don't know why it's named that way. This is another word that you're going to have to commit to memory. We do know it's a receptor organ, so that can get you the first part of the name, organ of Corti. So this is the actual structure responsible for auditory reception. And there are a couple structures I want to talk about on it, but you are not responsible for knowing them for the lab exam, but you just need to know the name of the structure. So we have hairs within our ear. They're called stereocilia, and they are sensory hair cells for transmitting sound waves. We can't see them super well on the slide, so I'm going to draw them in here. But what we would expect to see is these tiny, tiny stereocilia connecting the cells here, we've got 3 cells, to this membrane-like layer. This is called our tectoral membrane. And this membrane is going to attach to our hair cells, our stereocilia, so that when the fluid in the cochlea sloshes around due to sound waves, it's going to tug on these hair cells to create a sensory signal. And this is how sound gets transmitted. It gets converted from a sound wave to a sensory signal through this motion, and that signal is then sent to the brain for processing. So it's a really cool transformation done simply by the sloshing of that fluid and the tugging on these hair cells. So other things we would expect to see here, but that we can't see super well on the slide, we would expect to see nerve fibers connecting from our organ of Corti and leading back to that vestibulocochlear nerve. So those are just things that can help you figure out exactly what this organ is doing. But in terms of what you need to know for the lab exam to identify, you need to know organ of Corti, and you need to be really comfortable with this view. So again, we have 3 scala, 3 chambers. The middle chamber is really easy to identify, but you need to use these other 2 landmarks, the organ of Corti here, or this basilar membrane. You need to use those to identify your other chambers.